Hello everybody, I'm Mike Bishop. I'm a medicinal chemist working with Nanome, and today we're going to go in virtual reality and learn more about coronaviruses. And so today we've got an extra special guest with us all the way from Melbourne, Australia. I'd like to welcome Dr. Mike Kuiper. Dr. Kuiper?
Hello, thanks Mike, great to be here.
Do you want to say a few words about what you do?
Yes, so I'm a biomolecular modeler here at CSIRO, part of Data 61. And recently we've been working a lot on the coronavirus, particularly molecular modeling of the interaction of how the coronavirus actually gets into the cells, as well as a few other things about the coronavirus, which we'll be talking about today in VR.
Excellent. Why don't we put on our headsets and hop in VR? Let's do some virtual reality.
Fantastic.
Okay, what do we have here? Yes.
Fantastic, just getting myself here. Hopefully everyone can see that. So, yeah, so I want to just highlight some of the things we've been doing. Of course, this is not the first coronavirus we've seen. The COVID-19, of course, is quite a problem these days. There's thousands of scientists working on this right now as we speak. Lots of scientific research is coming out. But of course, there was the SARS coronavirus back in 2003. And actually from that information, from all the work that was done there, we actually have a good head start on this current coronavirus. And I'd like to discuss about that today.
Sounds great. You have some images of the SARS virus for us to look at?
Yeah, so over here, actually, so what I'll bring up here quickly is, so here's an electron microscope picture of the SARS virus. So this is from the Public Health Image Library. And what we can see here is actually the coronaviruses, a few of them there, 4 of them. And on the surface, you see this protrusion. That's actually the spike protein. So the spike protein is crucial in the coronavirus actually attaching to its host. So, and this is actually a model of the spike protein right here.
Oh, beautiful.
Yeah, so I just point out what we're actually doing here. So we're actually really interested to see how this protein attaches to the human cell. So based on a lot of work on the SARS coronavirus, we know that the SARS was actually binding to a particular protein called the ACE2 protein, the angiotensin converting enzyme 2. And it binds to these receptor binding domains, which we will talk about in more detail shortly, which are the little protrusions on the top of this model. So I should mention first a little bit why we're doing molecular modeling and molecular dynamics. We want to understand the function of these proteins in a much more realistic environment. These structures are beautiful, but of course, real life things actually move and wiggle and jiggle. So if I show you roughly what we're doing here, so this is just a quick diagram, which people can look at in detail later. When we simulate these, we make a model, and this particular model I made in Swiss Model, which is a publicly available platform which allows you to thread a sequence onto a template. And since we have a lot of star structures, we can put the sequence of the new coronavirus onto old templates and build a new model. And then what we do is we put this into a solvated solution, and we put into our protein, the ACE2 enzyme, and then we simulate this in molecular dynamics to see how the two will interact.
Oh, that sounds fantastic. Yeah, wonderful.
Yeah. So let me just put this away.
Okay.
So, of course, when we first started this, we were trying to do the whole structure, but that was a little bit too ambitious. So then we started to concentrate just on this receptor binding domain and the ACE2 protein. Now, there are a few important differences I should point out. Luckily, there was a paper just out yesterday in Science where the McLennan group actually solved the cryo-EM structure of the spike protein, and we'll bring that up shortly. But I just wanted to highlight first how different this virus looks compared to the SARS virus. So this is one of their figures, which highlights the differences in red of the binding surface.
And Dr. Kuiper, could you speak just a little bit louder, please?
Oh yeah, sure. I think I'm moving away from the microphone a little bit. So here we have an image from their paper in Science yesterday, which highlights the differences of the binding surface. In fact, what we can do now is bring up their structure and compare it to the model we made.
Oh, that'd be fantastic. Let's do it.
Okay, I'm going to delete this structure as well,
Okay.
just so we don't have the lag issues. So just coming in.
Okay.
Great, can you see that?
It's a little
Has
bit.
that come up yet?
It's a little bit behind for me.
Okay. Tell them to give us a few. I'll just wait.
Can you describe what we're looking at on that PDF there?
Sure. Okay, so this is the cryo-EM structure, which came hot off the press yesterday. And what we're actually seeing is a bit of a difference here, where the receptor binding domains are in different conformations. There's one up and two down. So, however, with the cryo-EM structure, we don't have all the resolution to fill in all the chains. So this is where molecular modeling can come in very nicely. So let me bring in the next structure from our actual molecular modeling.
Great. Oh, yeah.
So I'm just going to put this spike structure away for a bit and just focus on this. I'm just going to turn off the cryo-EM structure for a second. And I'm just going to show, hang on, I'm just going to tell these guys that. That's okay. We live in an open office here, so they come.
It's truly live.
So what we have here is the receptor binding domain at the bottom, and we have the ACE2 protein at the top. And this is actually part of a molecular dynamics simulation. So what I've done is I've taken snapshots of the simulation, extracted the good bits, and taken them into VR so we can interrogate the structure more closely. In molecular dynamics simulations. Did that come across okay to you?
It's coming up now for me.
Okay. So if I cycle through those images, we can sort of see that binding process as it occurred in molecular dynamics.
Okay. Dr. Kuiper, can you give us a minute to catch up?
Yeah, sure.
Yeah, so that's the ACE2 structure we're looking at. Yeah. Okay. Okay. So do you want to pull up the frames and walk us through?
Yeah, sure. So if I go back to the start, we can actually see the ACE2 protein on the top.
Right.
Is actually separated from the receptor binding domain at the bottom. So this particular trajectory only has 6 frames, but it highlights a lot of the key components of this. So if I step through this, we can see that it gets closer and then it actually binds. So this, I must say, is unguided, so there's no bias in these simulations. We just basically put these into a simulation and let them go and allow them to bind on their own.
That's really fascinating. Can we go back and look at the first frame again where they're separated? Yeah?
Yeah.
Okay, and the receptor binding domain of the spike protein, it looked like we saw some changes as it bound over in this region.
Yeah, we certainly will. And in fact, I have a workspace that we can open up later where I can show the differences between the SARS protein and the current COVID-19 receptor. But what I just wanted to highlight here is how this modeling can actually help with other work as well. So if we take the cryo-EM structure back, I'll just bring that back here. Let me just, what I'll do is I'll just change some of the views here.
Okay.
Just to make it a little bit more. I'm just going to turn off the atoms, just to give us a ribbon structure.
Okay.
And also with the cryo-EM structure.
Yes.
Just temporarily. So what we're seeing here is the receptor binding domain at the top, and you might be able to see that there's a lot of missing residues here in the cryo-EM structure. So when we take this, we can actually overlay that and fill in a lot of the missing pieces of the cryo-EM structure. So I think this is going to be quite valuable to give us a better understanding of what's going on at this particular point in time where we don't have a complete picture. So maybe together with a bit of experimental, a bit of simulation, we can see a clearer picture. And you can see how easy it is to make that in virtual reality. And I think this is the real power of doing this in virtual reality rather than on a two-dimensional screen.
Yeah, it's really remarkable also how close your model was to the actual cryo-EM structure when we look right through here. Really remarkable. Yes. Do you want to turn on surfaces, Mike, and we can look at the surfaces there, maybe look at the hydrophobicity?
Sure. So what I could do is I'll turn back a few of these frames. What I'm going to do is I'll delete the cryo-EM structure for now.
Okay.
To free up bandwidth. And then we can focus on this. So this is the ACE2 on the top. And what I'm going to do is I'm going to just bring it back to the first frame. And then we can create a surface from there.
Yeah, very nice.
So if I do the surface, and now if I color that, I'll color that with a hydrophobic surface. You can now look at these interfaces and how they might fit together. And you can see there's quite a bit of a hydrophobic patch over here. Would you like to take control for a bit, Mike?
Sure. Yeah, this is nice. Okay. Okay, I'm the presenter now. So I want to really get in here and look at this interface. And as you said, this yellow here shows us these large patches of hydrophobicity, and we can see them on both surfaces here. So that's exciting. And then I see some positive and negative charge regions as well, or partial charges. So there must be at least some really nice interactions here as this come together. I'm going to go ahead and run forward the frames, and we can see as it comes together. I'm going to go back to 1 and move it over. Yeah, we can see it start to come together there. Yeah, that is very nice. And what I really like in VR is just being able to get in here, make it very large and be able to get in and see these interactions as they're starting to form. I'll go back a frame and we can see just as it's starting to get in there close. Really remarkable. And I'll back out again and really get a sense for this. Yeah, thank you. Thank you for giving me a chance to be presenter, Mike. Please go ahead and take over.
Okay, grab that back. Actually, I'll just steal it. Right, I've got it back.
Yep. And feel free to talk. Feel free to talk loud, Mike, as we go.
Okay, yeah, sorry. I keep rolling back on my chair.
In excitement.
Now is I'm going to I'm going to bring up some new models. So I want to bring up an aligned model which is from the SARS virus.
And Mike, as you said at the beginning, we know the SARS virus also uses a spike protein to bind ACE2 on the surface of human cells to help its entry, right? To get started with entry?
Correct. Yes, so that's actually quite poignant in that. So what we'll do here is I've got two models up now.
Right.
One is from the SARS virus, from a previously sold structure, and one is our model of the COVID-19 structure.
Okay.
And what I'm going to do is just use some of these features that we have to actually highlight the interface.
Do you want to change the color of the SARS one, just so when you overlay them we can see the different ribbon colors maybe?
What I'm going to do is I'm going to just quickly, I'm just going to highlight that.
And Mike, I think we're still having a little trouble hearing you.
Oh, okay. Okay. All right, what I'm going to do now, these are separated, and I'm going to use a macro to actually highlight the interface.
Oh, awesome.
So now...
Oh, look at that.
So now this is where it gets really interesting, and this is where I think the power of virtual reality is so powerful for this sort of discussion. So here, I'll just have to remember which is which now. Let me just clean up a bit here. Do you want to take control there?
No, you continue as you do the alignment. I was getting up close and seeing some of the hydrogen bond interactions that we can see in virtual reality. It's really fascinating.
Yeah. All right, so let me highlight this SARS structure first. If we look at the SARS structure, we can see some of the hydrogen bonding interactions here and some of the hydrophobic interactions over here. In particular, you'll notice this lysine of the ACE2 and the tyrosine 23 here.
Right.
And if you look at the SARS virus, we actually have a tyrosine back here, nice threonine over here. There's a bit of hydrogen bonding to the backbone of the receptor over here. So hopefully that is clear.
Right.
You can get right inside this. And I can spend hours literally just poking around in here to see these sort of interactions, and it gives you a whole new sense of understanding, I think.
And Dr. Kuiper, do you usually like to look at it in stick mode or ball and stick or van der Waals envelope type of look? What do you like to do?
I like sticks. I have a personal preference for sticks because I think there's a bit more clarity there.
Yeah.
Sometimes if you try say a van der Waals, a lot is occluded, but
yeah, this is beautiful.
It really depends on what you're trying to look at. So now if I bring up close the coronavirus, I want to highlight some of the similarities and some of the differences here.
Great.
So like the SARS docking, we actually have the same lysine over here. We have the same conserved tyrosine on the receptor. But there are some notable differences as well. So we have this glutamine here, which is new. We have an arginine out the back here, which is new, and a glutamine here, which is new.
And can you see some of the interactions they're picking up?
Oh, yeah. So this is not the only simulation we ran too. We run these simulations multiple times, so we see more and more interactions as well. So just because we see one interaction in one simulation doesn't mean it's representative of all the interactions. So we like to repeat these sort of things over and over to get a more statistical ensemble sampling of the interactions. So the SARS virus, of course, was a static model where this is a molecular dynamics model, and what we're doing is seeing snapshots of that interaction.
Right. So I'm also, on the new coronavirus, I'm seeing a phenylalanine up here by this hinge where I think it might be, what is it over here, a leucine maybe?
Yeah, that's right. So there's a leucine over there. And in some of the other simulations which we can bring up shortly, we see additional, in another simulation, I actually have this lysine over here, which is new in the coronavirus, actually making a salt bridge.
Oh, yeah.
Aspartic acid over here of the ACE2.
Yeah, so that'd be a powerful interaction, yeah.
Yeah, so I will show that in the next model. I'll bring that up. So let me, I'm just going to delete this particular run and bring up another one, which is a slightly longer run. So it's 13 frames.
Okay.
When I bring these models into virtual reality, I often cut them down a bit. So I take out the waters, for instance, and that's predominantly to speed up the simulation.
Okay.
So what I'm going to do here is I'm going to just hide this other one, and I want to bring up this simulation here. So this is another simulation which I think is actually one of the more interesting ones, which we'll get into more of the details here. So this is a replicate of the simulation we saw before.
Okay.
What I want to do is highlight this the same way as we did the other.
Great.
I'm just going to quickly bring forward the simulation. Are you caught up yet? Is it working?
I'm seeing the structures, but I'm not seeing okay, now I'm starting to see the movement. Yeah, I'm seeing the frames now.
Okay, I'll just give it a moment to catch up. But what I'm doing now is basically the same as we did with the previous simulation. But this is an alternative binding, which is very similar to the one we saw before.
Okay, do you want to go back to the first frame and show us them separated?
Yep, so I'll bring it back to the first frame.
Okay, so great. Perfect.
Is that separated?
Yep. Yeah, definitely. I'm on frame 1.
Okay, great. Now if I bring that forward,
right, they come in. Oh, look at that.
We can see it binding. So what I'm going to do is I'm going to do the same representation as we did before to highlight the oh, actually I know what's going on here. Let me just quickly turn on this other structure.
Okay.
Get that out of the way. Let me try that again. That's better. Okay, so what we're seeing here are those interactions that I was telling you about before. So for instance, out the back here,
Mike,
we see that
it looks like you left the room.
We see that lysine here, which is now having the salt bridge interaction with the aspartic acid here. And if I turn this around, we can see this new glutamine here is having a nice hydrogen bond interaction with the conserved lysine of the ACE2. So if I just close this a little, you can see a little bit more detail here. Now, one of the more interesting things I think of this particular interaction is that we actually see a conformational change of the ACE2. So the ACE2 is, of course, an important enzyme in the angiotensin pathway. Hello, Mike.
Yes. Yeah, keep going, please.
That's okay. Yeah, I see you dropped out for a second there. That's okay. So I was just explaining here that what we're seeing here in this particular binding, which appears to be quite tight, that we actually see a conformational change in the ACE2 enzyme. So
hey Mike, could you
that
just
could be quite significant in that, of course, the ACE2 enzyme
Yeah, we're just waiting
is
for everything
involved
to
in
load on our end.
the hypertension pathway in converting angiotensin 2 to angiotensin 1 to 7.
Mike, could you hang on for a second while we catch up?
Yeah, no worries.
Thanks.
Mike, also ask the other mic to talk just a little louder.
And Mike, they'd love to hear you talk just a little louder on this end,
Okay.
if possible. I know you feel like you're yelling in an open space there, so it's tough. But thank you.
All right.
Thank you. Yeah, so, Dr. Kuiper, are you going to talk a little bit too about the recent results that appear to show a difference in binding affinity between the SARS virus and COVID-19 with ACE2?
You know, that's where we'll have to look a little more closely at the structure. Certainly, the atomic interactions are different to the SARS virus, and there's certainly a lot of hydrogen bond interactions, new hydrogen bond interactions that we didn't have before. So this is currently what we're looking at now, is to try and figure out just what are the details here which make this such a strong binder, as the current paper in Science has shown. It's actually shown, they've done some surface plasmid resonance experiments, which show that the binding of the COVID-19 virus to the ACE2 enzyme is about 15 to 20 times stronger than what it was for SARS. So that's quite significant.
So that could definitely lead to greater infectivity, possibly, right?
Possibly, yes. So there's certainly concern there that that's probably what makes it, or is a contributing factor to what makes it so such a tight binder and so infectious.
Right. Okay. Well, please continue. Yes.
Would you like to take control of this, or?
I'm right here behind you now. Sure. If you want to give me a presenter, that would be great. I'd love to get an up close look at it with my own hands. Let's see. I'm going to steal presenter. We are hearing a little background noise from.
I'm just going to just ask if it can be a bit quieter. Just
Sure.
a second.
No, that would be great. I'll go ahead and take presenter while you're doing that.
It's all right. We got them.
And then, Mike, could you talk about
How you doing?
what kind of insights contribute to a vaccine?
Yep. Yeah, it's just, Mike, do you want to give me presenter?
Steal present.
Yeah, it's not stealing.
Can you steal it?
I'm trying to steal it. I think it's just a little slow in stealing.
I
But-
thought I gave it to you, but
Yeah,
let me.
that's fine. Why don't we continue? We were wondering if, yeah, now I'm the presenter. Thank you. That's great. I'm just going to grab I just really wanted to get in with my own hands and get up close and look at some of these interactions at the interface. You know, there's this histidine here that really seems to be right in the middle of a few different hydrogen bond donors and acceptors. So you can imagine that this is a key region here in the middle of the interaction. Really, really nice.
This is the powerful thing. I think we can share. We'll be sharing, of course, all this data, so people can take a look at these structures for themselves.
Yeah. Mike, do you want to take back control? And we were wondering if you could tell us a little bit about how this work might help to inform people who are developing vaccines, or at least help them visualize the results of their work.
Yeah, sure. I'll just bring back the structure here.
Okay, great.
Yeah, so of course, you know, these viruses are not going to be static. They're constantly evolving as well. So the more we know about these interactions, the more powerful, the more things we can actually do about it. There's, of course, the opportunity to maybe develop some new drugs that are specific for this interaction. There's that possibility. But also when the epitopes on the virus change, we can then perhaps use the modeling to inform how to create new vaccines, or just be aware of those implications.
Yeah, as a small molecule medicinal chemist, and having worked in peptides, it's easy for me to imagine designing a peptide that's stabilized that might mimic, this part of ACE2. But as the receptor binding area mutates, as we get different mutations, I should say, those kinds of peptides may or may not bind as tightly. So it's an interesting problem with viruses.
It is. So this actually brings me on to the next modeling we could do. So we've of course been also looking at the coronavirus protease.
Oh yeah.
Which solved just a few weeks ago as well. So I think it's worth having a look at that as well.
Yeah, that would be great.
So let me just, I'll delete these structures for now. And I'll bring up our protease structure, and I'll describe what's going on there. This is, of course, another simulation.
Yeah, and while that's loading, I'll just mention for people watching the live stream, if they have questions, they can go ahead and send them in, and we expect to have a few minutes for questions and answer at the end. So
yeah, we'll be.
So on my end, it's still loading.
I'm just going to quickly just change. Okay, so hopefully you guys will be catching up shortly.
Yeah, it's coming along. Yep, it appears to still be loading, but I'm seeing most of it. Yep.
Okay, fantastic. So let me describe what's going on here. So many of your viewers will be aware that just about 2 weeks ago there was a new structure, so 6LU7 I believe it was, the crystal structure of the COVID protease. So what we do is we again put this into a simulation, and we put in some small molecular drugs, and we can actually run that through a simulation as well.
That is great. And are these small molecule drugs ones that inhibit related proteases, other viral proteases?
Correct. So this is actually lopinavir, which is actually a HIV drug.
Oh, right.
And people have been trying it for the coronavirus, and I'm not sure of how effective it is. However, it's a starting point to investigate, see if there's something else going on. So I don't know how that is playing for you. I'm just playing.
Yeah. Please continue. And this, and so it seems like virtual reality is extremely useful for seeing how these models look once you've been able to work with them a bit.
Yes. Yeah, I think what I love about this is being able to interrogate the data and actually get right inside and look at the actual details. So, for instance, here is a docking post. It's not particularly a representative post.
Sure.
But what I can do now is I can also highlight the rest of the structure.
Oh, that is so nice, yeah.
Yeah, do you want to drive a bit of that? No.
Sure.
So, of course, we've only just started this particular work, but this is one particular way where visualization in virtual reality can really help us understand the key molecular details of how a small molecule interacts with an enzyme.
Yeah, do you want to turn on the actual residues maybe in stick mode, and we can we can look at least where in this pose they've fallen there. Yeah. See if there's a...
Oh, you're the presenter now.
Okay, great. Let's see, I'm just looking in here, trying to get up close and look at these interactions. Again, this is really amazing to see. And so this is just one of many docking fits that you're going to find as you continue to do your simulations, right?
Correct.
And
It's really quite a dynamic situation, so those poses that we see in this aren't representative at all of an actual full drug dock. However, it does give us some ideas of how these molecules can interact with protease.
oh, wow. Okay, I'm going to go ahead and turn a surface on here. Let's see.
Very nice.
Yeah, I really like that. And let me see. Just going to look at lopinavir's actual structure there. Yeah, so I really look forward to seeing this when you've had time to run a bunch of different simulations and dockings and get to one that you are really excited about and see how it fits. But just being in virtual reality and having a chance to look at the 3D interactions up close is really exciting. I'll go ahead and pass it back to you.
Yeah,
And
fantastic.
Go ahead.
Yeah, I think we're almost time for some questions, I believe. Is that right?
I think it is that time.
So Mark's going to be choosing the questions, and Mark, you can just say them out loud to Mike, and they should be able to hear you. So.
Great.
Ready for some questions, Mike?
Sure.
Bishop?
Sure. Let's have them.
Question. One of our audience members wants to know if you can show the protease docking with some other molecules.
Mike,
Sorry, what was that?
Mike, do you have the ability, do you have the model ready to show docking with other inhibitors, other small molecules? And if not, why don't you walk us through the process you would use to quickly do that?
Sure. So when I do my simulations, we have to build in that set of molecules. So it's not as straightforward as just doing that. The sort of simulations we tend to do is sort of a brute force approach, where we put the protease in an aqueous environment, and we put in a small molecule to try and simulate without any bias, I should add. We allow the protein molecule to go through its dynamic flexing, and that way you can open up a lot of cryptic sites, as we know, that aren't actually obvious on first glance of the structure. So that's why I like to do a more detailed approach. However, that's computationally quite expensive, so we can't do the typical screening. So it's much more of an iterative approach. But first of all, you find something that you know docks, or we have some experimental evidence that shows it does dock. And then from there, you take it to the next level and you may make some tweaks to that structure, or do a more fragment-based approach where you may look at some fragments which are representative of the drug molecules that you might be interested in.
And Mike, again, that's trying to do an unbiased approach where you're hoping to uncover some cryptic sites, but one could, knowing where protease inhibitors typically bind on a viral protease, one could fairly straightforwardly dock in a biased sense known inhibitors, but that wouldn't necessarily give you the answer you're looking for. Is that correct?
No, and typically this is what we do. Once we see a hit inside a simulation, we can take it into virtual reality and delve in really closely and get quite intimate with the binding site. And by being in there, you can see how the interactions, how the molecules interact with the surrounding environment. and in particular how the residues of the protein are interacting with that molecule as well, and do the design from there. So it's a very powerful technique, I believe, to do it that way.
So Mark, today we can't show others, but one could certainly do that.
Yeah. It's a bit difficult to do it on the fly like this. I don't have a library available, but we can certainly separate out those molecules. For instance, I could actually do it. Let me just separate out a molecule. So what I'll do here, I'll actually just look at one of these molecules.
Okay.
So I selected one here. And what I'm going to do is I'm going to split that. So it will become its own entity, if you like.
Okay.
So if I've done that properly, now I can grab this as its own separate molecule. So hopefully you can see that. And now I can actually play around with that. And I believe some of the newer features will be able to visualize clashes as we're moving these molecules into the active site.
Yeah, and
We modified these on the fly as well. So this is really quite beautiful here. So I like this where I can take this molecule, I can take both the ligand that I've just pulled out of the active site, I can interrogate that active site, and if I want to, I can align them again, where it comes back together like that.
oh, that's nice.
Now they're locked together. So I really like that ability as well.
Yeah.
To basically interrogate this particular molecular interaction at that level of detail. So
Yeah.
just
Yeah, and if you did,
very intuitive as well.
Dr. Bishop, we have another question for you.
okay, we have another question coming in, Dr. Kuiper.
So yeah, for Dr. Kuiper, how often do you use virtual reality to look at structures like the coronavirus?
Funny enough, I actually use this pretty much every day now. I just find it such an addition. To use virtual reality to look at the structures is such a valuable addition to my work. I find it really compelling. In fact, I'll tell the story. I was a bit of a skeptic of VR originally. About a year ago, someone told me, you should get VR to look at your molecules. And I said, why would I do that? I can just do it on my screen, it's fine. I managed to get it installed on the computers at work. And it was so compelling. The next day I went down to the department store to buy my own kit at home, a Vive. So I've got it all set up at home as well. I just love it. It's a fabulous addition to what we do.
And how much better has VR been compared to your traditional 2D interfaces?
I think what VR does, and I think you have to really experience it to believe it, it allows you a very tangible interface where you can intuitively grab the molecules and manipulate them in a way which doesn't confuse your brain. Sometimes if you're looking at a two-dimensional screen, you get sometimes a bit of a flip, a bit of a ambiguity how the molecule is actually turning, and that can confuse your brain a little. But seeing it in true VR is just astounding to the level of detail. But you also, what I'm noticing is you're actually noticing a lot more interactions that you wouldn't have normally. So when you look at something on 2D, you often miss those things that are not your immediate point of focus. But when you're in 3D, you see it all.
Dr. Kuiper, I think people would be excited if would you be able to share your file with lopinavir, or is it not to the point yet where it's shareable? Yeah? Okay.
Yeah. Look, that's a known drug. This is really just some exploratory work. We can do some further modelling. Look, I think that would be good for the community impact as well if people want to contribute to this. So even improve some of the models, we'll make the models available as well. So part of the coronavirus spike protein, for instance, is very glycosylated. And that's one of the things I haven't included yet in the model because it's quite complicated to do. But I think eventually those are the things we want to include as well to make it as accurate as possible.
Yeah. Another thing, it was, it's always great. You're in Australia today, and I'm in California in the United States, and yet when we're in virtual reality, it literally feels like we're right next to each other, and we can hear each other like that. And I think that you have some ideas of how you can use virtual reality to collaborate, almost to crowdsource, if you will, in virtual reality. Could you tell us a little bit about what your hopes and dreams might be for that?
Yeah, I think this is a very powerful platform because, as you say, I think the presence that you feel, especially when you get a good connection, it feels like the person is actually in the room. I think the way the audio works, where it's directed, it's position dependent, makes it feel like that person is actually standing there next to you. And I think that is a very convenient way to do a lot of this work. I mean, especially because a lot of the researchers are spread all around the world. And this is a very powerful platform, if you like. And virtual reality is a very powerful platform, I feel, to engage on that level.
Have you had people join you in a virtual room that you didn't know before, that you've started to work with since you've gotten into virtual reality?
That was funny. I was actually at my dad's place, funny enough, a few months ago, and he lives in a very remote location, and I was actually using an Oculus Quest. And the funny thing was, I was just showing my dad's friends just what I do, and I noticed there was a room open that I thought was someone else I knew, and I jumped in there. And here I am in Australia on a remote island, phone connection, and I meet this guy in New York, and we have this great discussion about protein structure. So and that was just remarkable to have that sort of connection.
Yeah, that's fantastic.
So that was 10,000 miles away.
And I'm sure a number of people who are viewing right now are really familiar with virtual reality hardware, but if you're not, what I was on today is an Oculus Rift, but the Quest that Dr. Kuiper mentioned is a portable headset. You don't have to be hardwired to any computer, and it comes in a little case, and you can take it really anywhere you go, even a very remote island in Australia. And I also have taken the Quest overseas and used it at places where I only had my phone hotspot, for instance, for Wi-Fi. So it may not be quite as powerful yet for VR as the ones where you're hooked up to a desktop, but it's certainly great to be able to go in and do visualization really wherever you are.
Yeah, I found that too, especially working remotely. For family reasons, I had to work remotely, and it was great to bring a Quest along to quickly poke around at the structure. Of course, yes, the computational power is not quite the same as the desktop, but it was sufficient.
Yeah. And one more question, Mike, we have. So we've talked about visualization in virtual reality, and that's what we've been doing today, but my guess is you still do a lot of your heavy-duty computational work with other programs and then bring it into virtual reality to visualize it. Can you tell us a little bit about that workflow?
Yeah, so of course a lot of the simulations that we run are actually quite beefy. So some of the simulations that I showed you today were actually about 600,000 atoms in one case, which is the large spike protein, or 120,000 in the cut-down version, which is still quite a lot. So we use programs like NAMD for molecular dynamics, and we run those on large clusters utilizing GPUs. So that allows the efficient computation of a lot of these simulations, and for our simulations, the longer they run and the faster they run, the better for us, because it allows us more of that statistical sampling and more understanding of how the generalized motions are going. Of course, the molecular world moves very fast, very rapidly, and when we simulate these things, we're only seeing a fraction of the time that we'd love to see. So we're only seeing the nanoseconds, hundreds of nanoseconds or milliseconds if we run them long enough. But that's always a challenge. But as you know, computers are getting faster, so it makes this part of the work so much better. And I think, exciting. So typically, yes, I routinely take snapshots from those simulations and then bring them into virtual reality to do the detailed analysis.
No, that's great, and I can imagine as we go forward, you'll be able to bring bigger and bigger files into virtual reality. I know our live web stream is going to come to an end in just a few seconds, so on behalf of Nanome, I'd really like to thank you, Mike, Dr. Kuiper, for joining us from another continent and teaching us about coronaviruses and spike proteins and how they're so important to infectivity and how this type of work could really help us discover new drugs, new vaccines, and really push us ahead in our fight with coronaviruses. So thank you so much.
My absolute pleasure. And thank you for building a wonderful platform for this to make it all possible. And all the other hundreds of researchers out there which make the data freely available, because without that community support, none of this would happen.
Yep.
Thank you very much.
I look forward to our next visit in virtual reality, Mike. Thank you. Bye-bye.
Take care.